Hello everyone, this is Rohan Mishra, and here onwards we shall start discussing various concepts in phylogeny at finer details. If you directly end up on this video lecture, I would highly recommend you to first complete the previous few videos that are aimed to provide you the bird's eye view of the overall phylogeny concepts and their application. The upcoming videos are detailed discussion of the concept that are introduced in the previous video. So let's begin. Let us stick on to the distance method for phylogeny inference because you have already begun with it in our previous videos. We will cover character-based methods like parsimony and maximum likelihood later on in the upcoming videos. If you remember, while we were doing hands-on session on calculating trees in previous few videos, one of the very first step was to compute the genetic distance. I'm talking about this protocol. So we have followed this protocol, and one of the very first few thing that we need to do is that from the starting multiple sequence alignment, we first computed the genetic distance using any softwares like ProtDist. We have used ProtDist because we had protein sequences, and if the data contain DNA sequences, then you might have used this DNA test. So the overall idea is that when you begin in phylogenetic reconstruction, what you usually do is that you first need to uh, clear, or you first need to find out genetic distances among the sequences based on the multiple sequence alignment that you have. So for this purpose, in our previous demonstration, we have used this ProtDist program. So this ProtDist program provided us with the evolutionary distances in form of distance matrix. We have generated kind of 1000 distance matrices in our previous session. Now let me show you as to how this ProtDist interference looked like back then when we used it. Now if you focus over here on the very first option, that is option P, the very first option, the option P that is offered in this menu enables you to use an appropriate evolutionary model for your distance matrix calculation. So this ProtDist uh, provides, or you can say this ProtDist offers evolutionary models like JTT, PMB, PAM, Kimura, and categories model. So you have choice of using these different model for calculating the distance matrices in your study. In our case, we have simply used the default distance matrix, that is JTT, Jones, Taylor, and Thornton matrix, right? So using this JTT method, we proceeded for calculating our distance matrices, and eventually we generated the phylogeny. Uh, well, uh, we will look into more details of such evolutionary models, but for nucleotide sequences in this and upcoming few lecture. So now, if you open the DNA dist program that is used to calculate genetic distances in DNA sequence, here some different evolutionary models are offered. Like you can see that you can use this F84 evolutionary model for genetic distance calculation. You can use this Kimura model, Jukes-Cantor model, or log-det model. We will focus our discussion on these nucleotide substitution models here because we already discussed protein substitution models like PAM in previous lecture video. If you have not seen those videos, then you can watch them on the other playlist named Scribbles in Sequence Analysis on this same channel. The link to the playlist is provided in the description box below. Here we will talk in detail of F84 Kimura Jukes-Cantor model, evolutionary model in nucleotide substitution. And of course, we would discuss a few more complex evolutionary models as well. So now I hope that you get the context as to what exactly we are about to discuss in the upcoming lecture, and where exactly will it fit into the overall phylogeny protocol. So in short, we are going to deal with this step in your phylogeny protocol, and what are the different mathematical models and statistical details of those evolutionary model that will enable you to generate genetic distances between the given set of sequences. So this is what we are going to talk in more details in the upcoming few videos. Okay, so before any further discussion, it would be better if I introduce you with the concept of molecular clock. As we will be referring to this concept a lot of time in the upcoming lectures as well. So let us first get familiarized with the concept of molecular clock. So the story begins when Zucker-Candle and Pauling published two fundamental papers on the evolutionary rates of protein. Everything begins with this word, evolutionary rate of proteins. And these classic papers were published back in 1962. The citation to these papers are provided over here in this corner of the slide. You can read these paper for more details. So keep tab of the citation or sources if you want to dig in more details. So these guys, Zucker-Candle and Pauling, noticed that the genetic distance of sequences coding for the same protein but isolated from different species seems to increase linearly with the divergence time of the two species. So let me write this thing and then I will explain you what exactly do we mean by this statement. So the statement is the genetic distance of sequences coding for the same protein but isolated from different species seems to increase linearly with the divergence time of the two species. Okay, so this is what the main observation of this paper was. I will quote that again. The genetic distance of sequences coding for the same protein but isolated from different species seem to increase linearly with the divergence time of the two species. Now let me explain what this means. Say for instance, if you take example of maybe hemoglobin. Okay, so now if we take example of hemoglobin from different species, right? Okay, so if you continue with this example, if you take example of this hemoglobin and if you try to plot a graph that represent time on x-axis, okay, We'll say, for example, on x-axis, we have over here time in million years, 100, 200, 300, 400, 500, and 600. So this time is in million years. And if you plot on y-axis, let us plot substitution that happened in different hemoglobin sequences from various species. Well, let us say we are plotting substitutions. Okay, so if we plot time on x-axis and the number of substitution that might have happened in hemoglobin from different species, you might get a very peculiar graph. Okay, so you will find that the number of substitution on y-axis increase linearly as a straight line. You will find that the number of substitution on the y-axis appear to be a straight line that increased linearly. That is, as the time progress, the number of mutation that accumulates in linear fashion. With the passage of time, the number of mutations will increase, and this mutation and the accumulation of this mutation is in a linear fashion. So let me show you the real graph that was published in that paper. So you may note over here, for example, you may note that in case of mammals, just let us focus on this much portion of the graph of hemoglobin. In case of mammals, the number of amino acid substitution is less. Well, say for example, if you interpolate, you might end up like something over here, right? There were, say, some, below 100, below 20 substitution when it comes to the evolution of mammals. Okay, so here is what I want to explain over here is that you may note that in case of mammal, the number of amino acid substitution is less, that is around some 20, because their common ancestor was separated very recently in the evolutionary history. We know mammals are very recent in evolutionary history as compared to the life that originated far long back ago. Now, as the time increases in evolutionary history, the number of mutations also increase linearly. Note that the line increases linearly, this line of hemoglobin increases linearly till we trace the earliest vertebrate that shared the common ancestor, somewhere around 600 million years ago. So now there are several other proteins that show similar behavior. This was in case of hemoglobin, but along with hemoglobin, these people also identified that similar trend of mutations or amino acid substitution has happened with different proteins as well. For example, a very similar trend is observed if you plot mutation in case of cytochrome C and fibrinopeptides. You might observe that the linear lines for hemoglobin, fibrinopeptides, or cytochrome C, you might observe that these lines may have different slope, right? These lines may have different slopes, but they grow linearly for sure. So from this observation, one can conclude that mutations happens at slower rate in case of cytochrome C, and the substitution might have happened at higher rates in case of fibrinopeptide. Now, based on these observations, Zuckerkandl and Pauling propose the rate of evolution for any given protein is constant over time. That is what they concluded. They concluded that the rate of evolution for any given protein is constant over time. Now, this suggestion implies that some sort of molecular clock exists, right? If this is the time that rate of evolution for any given protein is constant over time, then this observation eventually implies that some sort of molecular clock exists. This molecular clock can tick as like other clock. This molecular clock can tick, while this clock can tick at various rates for different given genes. Okay, so this molecular clock can tick at a faster or slower rate for different genes. So however they tick, this ticking happen at a more or less constant rate for any given gene among different phylogenetic lineage. That is the main observation that Zuckergandel and his group proposed. Well, let me rephrase it again. The observation that the rate of evolution for any given protein is constant over time, this observation led them to suggest that molecular clock exists. These molecular clock can tick at faster or slower rates for different genes. However they tick, this ticking happens at a more or less constant rate for any given gene among different phylogenetic lineages. That is the basic concept of molecular clock. Well, now, introduction of this concept received an enormous popularity almost immediately after several reasons, right? We will discuss some of the applications of these molecular clocks here. So, the one of the very first application that can be that can be inferred using this molecular clock concept is that if a molecular clock exists and the rate of evolution of a gene can be calculated. This is the very first example. If such molecular clock exists, then the rate of evolution of any gene can be calculated. With this information at hand, one can easily use it for dating the unknown divergence time between two species just by comparing their DNA or protein sequence. Right? The second application is that if the information if the information about divergence time between two species is known, then the rate of molecular evolution of a given gene can be inferred. So this can be another application. If the information about divergence time between two species is known, then the rate of molecular evolution of a given gene can be inferred. The divergence time between two species can be estimated from fossil data. Third application, as you can say, we can assume Well, if we assume that such molecular clock exists, then drawing phylogenetic reconstruction become more straightforward. It becomes more easier and more accurate. Another application is drawing phylogenetic reconstructions becomes more straightforward. Right. So based on the introduction of concept of molecular clock, owing to these various reasons, this molecular clock hypothesis immediately got very uh, enormous popularity in very less time. Okay, so now let us come back to our topic of evolutionary model and try to understand how molecular clocks are relevant in generating evolutionary model. It is clear now that the very first step in the phylogenetic analysis of nucleotide or amino acid sequence is the computation of the matrix of genetic distance. In order to infer phylogenetic reconstruction, what you need to have first is that you need to have genetic distance, or simply sometimes people also refer it to as evolutionary distance. So what you need to have is the genetic distance or evolutionary distance between all pair of sequences that you are analyzing. Let us first talk about what are these genetic distances. Let us talk about what are genetic distances and how is that they differ from the dissimilarity matrix. These dissimilarity matrices are generated during multiple sequence alignment when we calculate that guide tree or dendrogram. So before we go to understand the concept of genetic distance or evolutionary distance, first let us try to define what is genetic distance and how is that it differs from the dissimilarity matrix that we generate from multiple sequence alignment. Well, now it is said that the DNA sequences are under continuous evolutionary pressure. That is, various evolutionary forces continuously act on DNA sequence and the sequences change by mutations. And these changes may be selected by natural selection if they are not deleterious. Usually in evolution, one ancestral sequence diverges into two daughter sequences. For example, in ancestral sequence that is shown over here, this is the ancestral sequence. In ancestral sequence, mutations happen in all of these position. You can see that mutation happened over here, mutation happened over here. It happened in the last two position as well as over here. So this we say that mutations happen in such ancestral sequence, or you can say ancestral protein sequence or amino or nucleotide sequence, and these happen independently in two lineages, right? By accumulating such sequence mutation, we say that the sequence has diverged. Well, say for example in this lineage, this C got mutated to T, right? This maybe C got mutated to G. In sequence 2, independent mutation happened and this A got mutated to T in sequence 2 you have got this G, where in original sequence there was T. This CA was mutated to TT, and we say that these mutations happen independently of each other. So by accumulating such sequence mutation, we can now say that the sequences have diverged from a common ancestral sequence. Right? So this is what I want to say. This figure represent the phenomenon of divergence. Okay, by accumulating these mutations in two different lineages, we say that these two sequences have diverged from the ancestral sequence. In the above case, we can say that the two sequence 1 and sequence 2, are derived from a common ancestor, and these two sequences have evolved independently of each other, and in due course of evolution they have acquired their own mutation. And based on this fact, finally we can conclude that this genetic distance is a measure of divergence, or in other words we can say a measure of this divergence is generally referred to as genetic distance. So what is genetic distance? It is nothing but measure of divergence between any two given sequences. Well, now this quantity plays an important role in many aspects of sequence analysis. For example, by definition, genetic distance provide measure of similarity between any given two sequences. Now, if a molecular clock is assumed to exist, then the genetic distance will be linearly proportional to the time elapsed from its divergence from the ancestral sequence, right? Now, let us bring in the concept of molecular clock. Now, if we say that molecular clock is assumed to be existing, then the genetic distance will be linearly proportional to the time that has elapsed when these two sequences diverge from the common ancestral sequence. That is, in other words, passage of more time leads to more divergence, right? If more time is passed, that would eventually mean that the two sequence will have chance to get mutate oftenly, and more divergence will happen between the two sequences. So now you know what molecular clock is, and we have defined the genetic distance as well, and somehow we have correlated them with each other. Well, again, for our subsequent discussion, we would have to have another set of assumptions as well. We have to take general account of uh, another assumptions, like we know that mutations are random event. Well, in general, it is believed that the substitution of nucleotides or amino acid in a sequence is a random event, right? So mutation can happen at any place. It is a random event. That is, we have to model it as a random event in any algorithm. Therefore, we need to have some statistical description. We need to have this statistical description to manage this stochastic process. Since it is a random event, we call it as a stochastic process. Now, if you want to model this random mutation event, then we must have certain statistical or mathematical description using which we can implement this concept into our algorithm or in our evolutionary model. Here onwards, we will try and understand the mathematical framework to model this process. So what would be our objective? Our objective would be to understand the mathematical framework to model the process of nucleotide substitution. Right. Our aim will be to discuss some popular evolutionary model that are used to describe such substitution of nucleotide or amino acid and understand how they calculate genetic distance. Right. We could discuss some popular evolutionary models, and our objective would be to understand how they calculate genetic distance in those evolutionary models. Our focus will be on nucleotide sequences over here. Okay, so now I hope you understand that since mutations are considered as random event, we would need to have some statistical description to manage this stochastic process. We will of course need a mathematical description to implement the concept of mutation as a random event in any algorithm, right? So this is what I want to convey from this slide. The next thing that you should know is that there is difference between what changes or what differences you observe in the sequence and what actually happened in those sequence in due course of evolution. So the simplest approach to measure the divergence between two strands of aligned DNA sequence is to count the number of sites where they have differ. For example, let us go back to the slide where we already did this in one of our previous video. Now, suppose we have these four nucleotide sequence with length of 20 residues. Okay. In this example, the divergence is expressed as the number of nucleotide differences per nucleotide site for each sequence pair. Right. So how is that we can calculate divergence between these two sequences, say sequence 1 and 2? We would have to figure out as to how many positions are there where mutation has happened. For example, sequence 1 and sequence 2, there are 20 nucleotides and they have four differences. This is the first differences. This is the first difference where in one sequence you have C, in another sequence you have A. This is the second difference where in one sequence you have C, another sequence you have A. At third difference there is G in first sequence and C in another sequence. And this is the fourth one wherein there is T in first sequence and A in another sequence. So in order to figure out the divergence between these two sequences, you can easily figure out simply by dividing the number of position where substitution has happened. Divide this number by the total number of or you can say nucleotide length of your uh, sequence as such. Okay, so if you divide 4 by 2, you would end up with 0.2. That will give you an idea of divergence. This is what I want to say. For example, if sequence 1 and sequence 2 are 20 nucleotide in length and have 4 differences, that would correspond to the divergence of 4 divided by 20 is equal to 0.20. So 0.2 is nothing but your divergence between these two sequences. We refer such divergence as simply observed distance. And sometimes this observed distance is also referred to as p distance, right? And this p distance is expressed as a number of nucleotide difference per site. We have got 4 sites, therefore you have got the observed distance or p distance. Well, now this p distance is very perceptive, or you can say it is very spontaneous measure to account for divergence, but unfortunately it suffers from severe shortcoming. We can say that if the degree of divergence is high, then p distance are generally not very informative with regard to the number of substitution that actually occurred. Well, let me write it down so that you would understand what is that I just said. I just said that if the degree of divergence is high, this p distance are generally not very informative when it comes to the number of substitution that actually occur during the course of evolution. This is you can say one of the biggest drawback of using this p distance. Say if the degree of divergence is very high, that is if you are trying to compare the two sequences that you know have common ancestor that has diverged a long way back in evolutionary history, in such cases the observed distance will not be very informative when it comes to the number of substitution that might have really happened in the long history of evolution. That is what I want to say. So let me explain this with an example. Suppose we have sequence of say cytochrome C, we have say sequence of cytochrome C, right? We know that this cytochrome C gene is retained in life form from the very beginning of life on our planet Earth. Say we have cytochrome C sequence from, say for example, we have got this cytochrome C sequence from, this is from plants, this is from yeast, this cytochrome C sequence is from fishes, another sequence is from cephalopods, we have another sequence from echinoderms, we have got another cytochrome C sequence of birds, and maybe from mammals. Right. Well, let me denote it. This is cytochrome sequence from plant. This is from yeast. This is from fish. This is from arthropod. We'll see, for example, in a cephalopod. This is from echinoderm, and we have one from birds and another from. Okay, and let us draw an elementary evolutionary tree. This is just say for example. Right. So this is what we have. In this particular example, we have cytochrome C sequences from different genera that correspond to various life form on our planet Earth. Well, now we might observe that there is difference of, say for example, A in plant, right? In, well, for example, at this position in cytochrome C, you have got A, and we have G at the same position in yeast, right? There might be T in somewhere over here in mammalian sequence, and there might be C in bird sequences. Okay, and so on. The idea is that there might be differences in amino in at these positions. So now the differences that we observed in these sequences are not the same as the number of changes that have actually occurred during the evolutionary history. So whatever differences that we observed here are not the same, differences that might have happened in these millions of years of evolution. That is what I want to say. Let me write it this once more. The differences that we observed in these sequences are not the same as the number of changes that have actually occurred during the evolutionary history. As you know that evolution is a very long process, and it took millions of years and hundreds and thousands of generations to bring about these changes that we observe now. There is a long sequence of changes that might have taken place in this evolutionary history that is simply not evident now by simply comparing the sequences, right? Let us take a brief review of various changes that can happen in evolution. Okay, I will just give you a brief introduction as to what changes might have happened in evolution that somehow we cannot appreciate by comparing what are the changes that we see into the current sequences. Okay, now consider this situation. Consider a situation over here wherein in one sequence we see that there is A present, and in another sequence we see that there is C present. Okay, and these two sequences, of course, they have derived from a common ancestor A, and at the analogous position there was A, right? So here we observe only one change and assume that in the ancestral gene only one change might have happened, and thus we have C in sequence 2 and A was retained as it is in sequence 1. We call it as one change and one difference, and hence refer it to as a single change. So this is the simplest thing that one might expect. Now consider another example of two sequences. We have got, say, this sequence 1 and we have got this sequence 2, and of course we have got this ancestral sequence. So we'll say, for example, one from mammal, okay, and another from fish. So say the sequence with A, this is from mammal, and this sequence is from fish, right? We know that it was a very, very long time ago when mammalian ancestor separated from the fish. This happened a very long time ago, and it's completely possible that multiple changes might have happened in this period. But we observe only one change. That is, in one sequence we have A and in another sequence we have T. We might miss out an intermediate change C that happened somewhere in the middle, right? So similarly, it might have happened, that the sequence changed twice, right? The sequence might have changed twice, that is, two changes occurred, but we observed only one difference over here. We call such changes as coincidental changes, right? You can see that in ancestral sequence there was A, this A changed to G, and this A again changed to C in another sequence, right? Therefore, two changes have happened, right? Two changes have happened over here, but we see that there is only one difference, right? So such changes are called as coincidental changes. The changes can also be classified as parallel changes, and parallel changes are assumed to happen when two changes happen in the background. When two changes happen in the background, but since the sequence is the same, it appears that nothing has happened over here. Just consider this example. In ancestral sequence there was A. In first sequence it got changed to C. In another sequence it got changed to C, right? But since this is what we observe, and it appears that nothing has changed, but actually both of the sequences has got common ancestor where the nucleotide A was present. But now in both the sequences we have got C, and hence it appears now when we compare the sequences that nothing has happened. Such a changes are called as parallel changes. That is, in ancestor there was A, but in both the daughter species you have got C, right? So if there are three changes that happen and end result is no difference, right? In ancestral sequence, this A changed to T, intermediately this A changed to C, and this again C changed to T, right? If such is the scenario in which if there are three changes that happen and at the end, what is the end product? In both the sequence, you observe that there is T, right? So such changes are called as convergent changes as they appear to happen in the same line, right? So if there are three changes that are happening and at the end there is no difference, we call such changes as convergent changes as they appear to happen in the same line and the end result is the same T. Now another situation can be like, what if the change happened and in subsequent time it got reverted back to the original nucleotide? We call such changes as back changes. As you can see, in subsequent evolutionary time, A in the intermediate group it got changed to C. But again, the C was again reverted to A, right? And now it would appear that nothing has happened over here. Such a changes are called as back changes. That would result in two changes but with no differences. Convergent changes can be like there might be three changes but there is still no differences. There can be parallel changes where there are two changes but still no differences. There can be coincidental changes where you can observe that there are two changes but actually there is only one difference, right? In multiple changes there are two changes but there is only one difference. Well, these things usually happen in evolution, right? My point is that what changes we see now might be smaller than the amount of changes that might have actually happened during the course of evolution. And this is what nucleotide substitution models are for. These nucleotide substitution model will take into account of this underlying evolutionary event that might have happened in the past and compute true genetic distance. We refer this as D. We refer this as D, or what we call it as true genetic distance. Therefore, true genetic distance D correspond to the actual number of substitution per site that might have occurred. Therefore, we oftenly say that in this way, the observed distance P is underestimates the true genetic distance D. Right? So here is the difference between the observed distance that you might have observed in multiple sequence alignment and the actual genetic distance and evolutionary distance that might have really happened, the changes that might have really happened in long course of evolutionary history, right? So you can also somehow get a relationship between such expected genetic distance and observed genetic distance. Well, now if we plot the genetic distance d on x-axis and the observed distance p on the y-axis, a very characteristic graph might appear, right? So using such graph, one can easily interpolate genetic distance if observed distance is known and vice versa, right? So, well, say for example, if the observed distance is 0.23, right, then the genetic distance might correspond to something over here like 0.5. Or if the genetic distance is 2, then the observed distance, if you try to interpolate it, it can go to about 6.5, right? So in this way you can kind of interpolate between the genetic distance and observed distance, right? That is, one can estimate the divergence if the genetic distance is known. If you know the genetic distance, one can easily justify the divergence. This transformation is generally non-linear. This is this transformation or this relationship between the genetic distance and observed distance is not a straight line, but it is, you can say, you can see it is not a straight line, it is non-linear relationship. One interesting fact that can be inferred from this graph is that as the evolutionary time goes by, multiple substitution per site will accumulate and ultimately sequences will become random or saturated. So this is what I have just said. As evolutionary time goes by, multiple substitutions per site will accumulate and ultimately the sequence will become completely random, and we say that the sequence has saturated. Okay, so just infer this graph. As the genetic distance increases, the observed distance somehow goes on increasing, right? But when the observed distance comes around say some 7.5 or so, or when it crosses the limit of 7, whatever is the genetic distance, if you try to increase, there is no apparent increase in the observed distance. That is what we say when the two sequences have become absolutely random, right? And we say that the sequences has achieved saturation. Okay, so this saturation is observed here at say p value of around 0.7. So when the p value comes to 0.7, whatever genetic distance is increased after that, it would not result in increase in the p value or observed distance, right? This is what I want to say. The saturation is observed here at p value of 0.7, where there is no apparent change in the p value even if the genetic distance increases. Even if the genetic distance increases, there is no apparent change in the p-value, right? In such case, we say that the sequence has become random or acquired saturation. Well, now, this characteristic shape of this curve will depend on what substitution model you have used to plot the genetic distance and the observed distances, right? So this figure, this particular shape of graph, well, this graph is of Jukes-Cantor evolutionary model. Well, now let us conclude today's lecture uh, by by our conclusion that we can fairly agree on the fact that genetic distance cannot be observed directly, and therefore some statistical techniques are necessary to infer this quantity from the data that you have, right? What we can conclude from our discussion, that we can conclude that the genetic distances cannot be observed directly from the sequences that you have, and therefore statistical techniques are necessary to infer this quantity. This quantity, what quantity? We call it as genetic distance. To infer this quantity from the sequence data. This is what we can conclude from today's lecture, that genetic distance, well, it is completely different from the observed distance, and we would need some statistical model that would be eventually essential for calculating the true genetic distance, for computing the divergence between any two given sequences in our data, right? So I should stop this lecture for time being. In our next lecture, we would start with the evolutionary models as such. Okay, and yes, uh, you can refer this book if you want to go into more details of whatever we are going in this line of discussion, right? So this is the book that I have referred for calculating the evolutionary distance and studying the evolutionary model. You can use it or you can read it. It might be present in your library.